In our previous lectures, we've seen how our amino acids can link together to form these secondary structures. Right? We saw our beta sheets, where we have our amino acids linked together in these long strands, and we have our alpha helices, where they're coiled around each other. Now, of course, one of the main driving forces that we saw for these secondary structures forming was hydrogen bonds between our backbone of our amino acids, right? When I'm referring to backbone, I'm of course referring to my peptide bond, where I have my carbonyl as that hydrogen bond acceptor, and I have my nitrogen with the hydrogen attached as my hydrogen bond donor. Now remember, of course, my carbonyl here and my nitrogen are always present within my peptide backbone. So these sorts of interactions that we see in our secondary structures only require our actual peptide bond that links all of our amino acids together, right? So far, we've really seen very little role of our actual amino acid side chains in our protein structure. This changes when we look at our tertiary structure. So protein tertiary structure is the overall 3D structure of our protein. So we form our secondary structures, and then those will be combined together in order to form this overall 3D structure. Now, for our 3D structure, we're going to be looking primarily at our side chains, right? The backbone is mostly involved in the secondary structure, and now we're looking for interactions between our side chains to form our tertiary structure. So our tertiary structure will involve numerous weak interactions between amino acid side chains. These will be essentially all of our typical intermolecular interactions between different molecules, except in this case, it will be between different amino acid side chains within the same large protein molecule. So these sorts of interactions can then bring areas of our protein together that might be very far apart in terms of sequence. For example, you can see right here we have a serine near the start of our protein sequence that's hydrogen bonding with another amino acid much further along in our sequence here. So let's talk through some of these different interactions that we can see. So the first and one of the most important interactions that determines our tertiary structure is the hydrophobic effect along with Lund dispersion interactions between our nonpolar amino acid side chains. So as we've seen previously, the hydrophobic effect is based on this change in entropy that occurs when we release water molecules that are forced into an ordered structure around our nonpolar molecules, right? So if we can cluster our nonpolar molecules together, that allows us to release the ordered water molecules that are around those nonpolar molecules, and therefore it will allow us to increase our entropy, right? So we're trying to avoid this ordering effect of water. In the case of a protein, right, so if we have a chain of amino acids, in black here we have our polar amino acids represented. So this would be things like serine or lysine, those more charged or more hydrophilic amino acids, at least referring to the side chain again. And in white here, we have more of our non-polar amino acid side chains. So these might be things like leucine or our valine, things that have very large carbon chains as part of their amino acid side chain. Now, if we put this protein strand into an aqueous solution, which of course, most of our biological environments are actually aqueous solutions, right? If we're looking inside of our cell, our cytoplasm is filled with an aqueous solution with, of course, many solutes dissolved in it, but we still have a lot of water surrounding all of our proteins. So when this protein chain is exposed to that water, so as we apply our water here on the outside of that protein, all of these white hydrophobic amino acids are going to try to hide from that water in order to increase our entropy. So we end up structuring our protein something like this, right? Where all of our non-polar amino acids hide on the inside, and our more polar amino acids are exposed on the outside of our protein. So this is going to be one of the key driving forces that we see for a lot of protein structure, right? We're always trying to bring our nonpolar groups away from the water solvent that's surrounding our protein, and then we want to expose our more polar amino acid side chains. Now, of course, as with our secondary structure, we also need to consider hydrogen bonding. So within our amino acids, we of course have many different hydrogen bond donors and acceptors. Previously, we saw with our peptide bond shown here that again, we have our hydrogen bond acceptor of our carbonyl carbon and oxygen, and our hydrogen bond donor of our nitrogen and its hydrogen. Now again, of course, these are mostly involved in our secondary structures, our alpha helices and beta sheets, but they can also be involved to some extent in our tertiary structure as well. So then we'd be expecting to see more things like our serine, for example. So serine would have a alcohol group in the side chain. Threonine has an alcohol group as well, right? So alcohol is a very good hydrogen bonding with the hydrogen bond acceptor there with our hydrogen, or the hydrogen bond donor rather, as well as the hydrogen bond acceptor with that additional lone pair on our oxygen. Now, it is important to note that hydrogen bonds aren't always going to be the greatest driving force in protein folding, because we can also hydrogen bond with water, right? So our serine is perfectly happy hydrogen bonding with water as it is hydrogen bonding with, say, a threonine within our protein. So they can contribute to the overall free energy of folding, but they don't in and of themselves tend to determine the folded structure. The next key interaction is electrostatic or ionic interactions. So electrostatic interactions are long-range, strong interactions between our permanently charged amino acid side chains. So here we'll be seeing all of our key negative and positively charged amino acids. So we have aspartic acid, or of course glutamic acid, or aspartate and glutamate. And of course, for our positively charged amino acids, we have things like arginine shown here, or our histidine and our lysine. So all of these amino acids can play a key role in forming ionic interactions or electrostatic interactions across our protein. So if we have differently charged groups on different secondary structures, right, we can bring those close together in order to form those interactions within our protein tertiary structure. All right, so we have a positive charge here that wants to interact with other negative charged amino acids that can be brought nearby. So these electrostatic interactions can end up forming salt bridges within our protein, where they're making essentially a bridge with this ionic or salt-based interaction between two different regions of our protein. Now, you'll note, of course, before that I mentioned that we are mostly focusing on putting our hydrophilic and charged residues on the outside of our protein, and we want our more nonpolar amino acids on the inside of our protein. However, when we have charged amino acids that form an electrostatic interaction, it can actually be helpful to have those inside of our protein. So all of our charge interactions are affected by something that we call the dielectric constant. So it relates to the conductance of a material, or essentially how well charges can be stabilized or can move around within that particular environment. Within water, charges are very happy, but they're very happy, right, because they have a lot of different things they can interact with, right? So we get this very high dielectric constant for water, representing that that high conductivity of our water and the ability of our charged particles to form many different interactions within that water environment. Within a more nonpolar environment, like an oil, you have a very low dielectric constant, right? It's not a conductive material. Our charges can't really move around within that material. And so what happens is if we have a charged interaction, it actually gets strengthened, right? Because these charges really want to stay together here. This is the only possible interaction that they can really have within this nonpolar environment. Otherwise, right, they're just kind of left all by themselves. In fact, if you remember Coulomb's law that we talked about when we looked at ion solvation earlier, the force of the attraction between two charged particles is based on the charges of those particles over the distance between them squared times this dielectric constant. So as our dielectric constant increases, the force of attraction between those two charged ions here of our amino acids actually decreases, right? So it gets much stronger in that nonpolar environment and much weaker in a more polar environment. Again, there's just more possibilities for other interactions, or we could say that there's more shielding of our two charges, right? They're too busy, they're too distracted by the other charges within that water-based environment. So just lowering this dielectric constant in the center of our hydrophobic environment in our protein will actually increase the strength of these interactions, as long as we can still maintain that interaction of the positive and negative charge, right? You really don't want a positive charge just by itself within that nonpolar environment. So a high dielectric constant will equal a weaker ionic interaction and a weaker salt bridge. A lower dielectric constant will lead to a stronger electrostatic interaction and therefore a stronger salt bridge. So far, we've only been looking at varieties of non-covalent interactions within our protein side chains. Now, there are also some covalent bonds that can form between our amino acid side chains in order to influence our protein tertiary structure. So the most important example of this is our disulfide bonds. So our cysteine amino acid has a thiol group in its side chain, and our thiols can actually be easily oxidized to form a disulfide linkage. So two of our cysteines can be linked together in this cysteine disulfide linkage. This would be through an oxidation reaction as we're eliminating those two hydrogens. And we then have a very strong covalent linkage between these two amino acids. Again, these could be very far apart in our overall protein sequence. And so you could then bring those two areas of the protein close together and link them semi-permanently there to each other to create a certain tertiary structure. Now, of course, the other thing with our disulfide bond is it is also quite reversible, right? We can oxidize it, but we can also reduce it and break that disulfide bond. So it is a semi-permanent covalent bond that will have a large effect on our protein structure, but we can also break it without too much difficulty if we have a reducing reaction. So all of these different factors will come together to determine our overall protein structure, right? So as we're kind of moving around to try to form these different interactions, it will lead to this overall spatial arrangement of our atoms within that tertiary structure. Now, in the rest of this lecture, we're going to look at some examples of what tertiary structures can look like for different proteins. So first of all, we're going to talk about some different kinds of tertiary structures. So we normally break our proteins down into a couple of different general categories. So we have more fibrous proteins, such as a collagen shown here. So as the name suggests, these tend to be more extended proteins in more shapes like fibers, essentially. And we have more globular proteins that are more compact and they're more glob-shaped, if you will. So globular proteins tend to be the most common proteins. They're the ones that tend to move around in our cells and do a lot of the different common functions that we think of proteins as having, such as acting as enzymes, for example, or even carrying different things such as oxygen in the case of our hemoglobin shown here. Our fibrous proteins tend to have more of a structural role. So they're more the proteins that are holding things together within our body. let's take a look at our fibrous proteins first. Now, one of the key things that we see for all proteins is that our structure relates very closely to the function of our protein, right? So we form some kind of 3D structure, and then we're going to use that specific structure to help us carry out some specific function. So for our fibrous proteins, since they tend to have a more structural role, we're going to be trying to form very tough and rigid structures or flexible structures, depending on the need, in order to make the necessary structural features that we need within our body, right? So we have fibrous proteins such as alpha-keratin involved in hair and feathers and nails, lots of different tough structural features. We have collagen in tendons and in the bone, again, to create some of those strong structures that we see. So we're going to take a look first at alpha-keratin, but we'll be looking at each of these three proteins to see kind of their key structural features and how that relates to their function. Okay, so let's start with our alpha-keratin. Here is an overall picture of a hair. So within our hair, of course, we have kind of the overall fiber of our hair, which is ultimately composed of cells. And within these cells, we have these filaments that are ultimately composed of alpha-keratin. So down here at the end, we have actually our alpha-keratin structure here. You'll notice it does have an alpha helical component, which we'll take a closer look at here. So here's the structure of our individual alpha-keratin protein. Alpha-keratin adopts almost entirely an alpha helical structure. So notice, of course, our typical alpha helix ribbon shown here. So we have this very long helical structure, but then we actually take two of our alpha helices and coil them together to make this two-chain coiled coil, right? So we have the coil of our alpha helix, and then we're coiling it together with another alpha helix to form an even stronger structure. Now, one of the interesting things to note here with our alpha-keratin is that it actually uses a left-handed coiling of our two alpha helices together, right? So remember, the alpha helix itself has a right-handed coil to it. So we have our right-handed alpha helix combined with this left-handed coiled coil. So it's kind of coiled together in opposite directions in order to create this strong fibrous structure. Now, once we have our individual alpha-keratin here, we're going to link these together in order to create those larger fibers that we saw in the previous structure. Now, of course, when we want to link these fibers together, so we're going to need some sort of strong interaction that can link these together. And within our alpha-keratin, we rely on disulfide bonds primarily. So approximately 18% of our amino acids are cysteines. Cysteine, of course, has that thiol that can bond together to form our disulfides. So within our alpha-keratin, we're going to have a large number of disulfide bonds linking our different alpha-keratin units together. So as we create those larger fibers that then end up making our hair, we have all of these disulfide bonds linking that hair together. Now, it turns out these disulfide bonds actually also relate to permanent waving or curling of your hair. So the particular pattern of your disulfide bonds within your hair will determine whether your hair is curly or straight. If you want to change that, you actually have to break these disulfide bonds and then reform them in a different orientation in order to create that curl, right? So whenever we break disulfide bonds, we first reduce it to form our original thiols. So here we have our cysteines and their thiol forms not linked together. We can then physically change the shape of our fibers here. So if you curl the hair up here, you're changing that shape. And you can do that, of course, because it doesn't have the disulfide bonds anymore. We can then use an oxidation reaction to reform our disulfide bonds, but in a new orientation, so that now we're locked into this different shape. So a lot of the agents that are used to create kind of the permanent curling or straightening, it's actually including reduction and oxidizing agents in order to actually change this disulfide bond linkage within our alpha-keratin. The next fibrous protein that we're going to look at is collagen. So alpha-keratin, of course, we saw this reliance on our alpha helices. Collagen is going to use a very different kind of helix. So collagen actually uses a new secondary structure. So it's a left-handed helix as compared to our right-handed alpha helix. And again, it makes up a key component of all of our connective tissue, such as our tendons and even our bones. So here's a picture of what that left-handed helix looks like for our collagen. So our collagen uses glycine, proline, and a hydroxyproline. So that's a proline with an extra OH group added on. So you can see that over here with the red of our oxygen. So notice, of course, we have two different amino acids here that we would normally call helix breakers. So this is rich in helix breakers. And our helix breakers, of course, are called that because they don't like to adopt that normal alpha helix configuration. But the very features that make them bad for an alpha helix actually make them incredibly useful here for our left-handed helix. So in this left-handed helix, we actually have a very tight helical structure. So rather than the 3.6 or almost 4 amino acids per turn in a normal alpha helix, we only have 3 amino acids per turn here within this helix. So it turns at a much sharper angle. It's also actually packed together with 2 other left-handed helices. So we have this bundle of 3 left-handed helices combined together. You can note that with our different colors here of those 3 different helices. So we get this very tight arrangement, and our glycine and proline are actually very helpful for allowing this tighter structure compared to our normal alpha helix. This hydroxy group here of our alcohol actually has a special function here. So normally with our proline can adopt 2 different conformations. We refer to these as endo and exo conformations. You might remember those terms from organic chemistry. So we have, of course, our ring structure that we create with our side chain, and it can either point up in this endo conformation, or it can point down here in an exo conformation. When we add this hydroxy group here, it encourages our ring to point down in this exo conformation to allow more hydrogen bond formation. And this then will help to promote that tighter helix structure that we see here in collagen. So our 4-hydroxyproline is a requirement for our collagen. If we are missing our 4-hydroxyproline, it actually creates a huge issue within our body, as we can't form our collagen anymore. So this is actually what causes scurvy. So in scurvy, of course, scurvy can be treated by vitamin C. It usually occurs when you have a lack of fresh fruits and vegetables in your diet that are providing a lot of those essential vitamins, such as our vitamin C. So as we go to form our hydroxyproline in order to make new collagen, we actually have to react our normal proline in order to form this hydroxyproline. So we take our proline and we use a proline hydroxylase enzyme to add on that OH or hydroxy group onto our proline. Now this reaction uses our vitamin C here as a key cofactor. So we have an oxidation reaction where we actually have an iron here that's in our enzyme itself. It's getting oxidized and reduced. And in order to regenerate that iron to continue our reaction, we have to have our vitamin C present here, right? So vitamin C gets oxidized as our iron gets reduced, and then our iron is ready to react again to create more hydroxyproline. So if we run out of vitamin C, if it's all consumed in this reaction, we're not going to be able to produce hydroxyproline, we're not going to be able to produce collagen, and gradually we're not going to be able to create the connective tissue that we need in order to hold our body together. So essentially, when you go into a scurvy state, you actually are kind of starting to fall apart in a sense, as you can no longer make that collagen of our key component of that connective tissue. So once we form our triple helix structure of our collagen, again, just like our alpha-keratin, we're going to combine that collagen fiber into larger collagen fibrils by cross-linking those collagen proteins together. So we take our individual collagen triple helices and we cross-link them. And we actually do this using a lysine or a hydroxylysine along with histidine amino acids. So it's a rather unique covalent bond that we form between our collagen units, again, to kind of build this larger fiber here. And again, these fibers can get combined together to make larger and stronger structures, and you get these quite large collagen fibrils that you can actually see under a microscope. This would be an electron microscope image here. The final structural protein that I want to talk about is silk fibroin. So silk fibroin uses our other secondary structure of the beta sheet. So we form these very large anti-parallel beta sheets. Remember, our anti-parallel beta sheets are also our beta sheets that have the stronger, more linear hydrogen bonds between our beta strands. So we have stronger H bonds there within each of our individual beta sheets. And then we're stacking our beta sheets on top of each other to create this silk fibroin structure. All right, so notice you have this beta sheet marked in red, stacked on top of our blue, and stacked on top of our green beta sheet. So all of these beta sheets are stacked on top of each other to form our overall protein structure. Now, in addition to forming this anti-parallel beta sheet, a silk fibroin structure relies primarily on alanine and glycine. Right? So notice we have our glycine and alanine amino acids. So alanine has the methyl group coming off, marked in black here. Glycine, of course, has only a hydrogen. Right? So this is the extent of our side chain here for glycine. Now remember, with beta sheets, our side chains are always pointing alternately up and down from our beta sheet. Right? So you can see how the alanines are kind of alternating, going up and down here as our strand is lying sideways here within this, this diagram. Now, of course, the advantage here of having alanine and glycine is twofold, right? So in one case, we have a lot of nonpolar side chains here, right? So we're going to create a hydrophobic effect, bringing our beta sheets closer together. And we also have relatively strong London dispersion forces as well. Now, our London dispersion forces are going to be strengthened by the fact that we have very small amino acid side chains here, right? So with only our alanines and our glycines, we're able to pack these beta sheets very closely together, right? Notice kind of how nicely these can sit together as they're resting on top of each other. We can create a very large surface area that then creates a much stronger London dispersion force interaction between those beta sheets. Right? One of the keys to creating a stronger interaction is creating a larger surface area for that interaction to take place. So our silk fibroin is all about creating that very strong or that very large surface area to then magnify the effect of our interaction. So our silk fibroin, of course, makes up spider silk. So one of the things that we know about spider silk is that it's an extremely strong material, right? We have those sheets lying on top of each other, creating a very strong interaction as we create that large surface area and we pack those sheets closely together. But the other thing that's unique here is that we don't actually have covalent bonds linking those sheets together like we did for our collagen or our alpha-keratin. So what this means is that they can really stretch a lot, and there's actually a lot of flexibility within that structure, right? We can take one beta sheet and we can line the other beta sheet up next to it. But if we pull it and one of our beta sheets slides, it can still interact with the beta sheet that as long as there's some overlap there, right? So it might even get pulled over to overlap with another beta sheet as well, and we can have interactions between those two beta sheets, right? So we can kind of have the sliding effect between our beta sheets where they can slide against each other if there's a strong force applied, and they can stretch, but they can still reform and still have those strong interactions between our beta sheets, right? So it's a very strong structure, but it's also a quite flexible structure as well. So far we've only been talking about the structures of fibrous proteins. Now again, fibrous proteins have very different structural features and structural needs compared to our globular proteins. Right? Fibrous proteins, they form extended structures. They're very strong structures. Our globular proteins, we want to clump up and form these smaller but more mobile structures. So our globular proteins are very packed together. So you can see that by this comparison here, right? So if we take a beta conformation of a protein and we just stretch it out all in beta conformation, we'd create a very long structure. So 2,000 angstroms, that's 10 to the negative 10th meters by 5 angstroms in width, right? So this very long extended structure. And that's essentially what a silk fibroin might look like, right? Just one sheet of that silk fibroin. If we twist this into an alpha helix, it gets a bit shorter, right? So now it's 900 by 11 angstroms, right? Because now it's coiling, so it's a little bit more compact, but still very extended structure, right? That would be, again, something like alpha-keratin. When we pack it into our globular form, notice how small and compact this protein is, right? It's now only 100 by 60 angstroms instead of 900 or 2,000 angstroms long. So we're taking that and we're packing it together in this little ball. And again, one of the key advantages here is that our globular proteins are usually in aqueous environments. So we're trying to hide all of our hydrophobic amino acids here. With our fibrous proteins, we often don't care if we have lots of hydrophobic amino acids because we're not trying to dissolve and mix in with water. We're trying to form a strong structure that's going to stay how it is without getting pulled along with our aqueous solutions. So globular proteins are more compact as we hide our hydrophobic amino acids. Fibrous proteins are extended and they don't really care about hiding those hydrophobic amino acids. Our globular proteins are going to tend to be a lot more diverse in terms of the structures that we see here, right? So we're not going to have just as many simple structures where we just have basically an alpha helix repeated through the whole structure like we see for our alpha-keratin. We do see some kind of shared similarities in terms of our structural features, though, and we call these motifs. So motif is a specific arrangement of several secondary structures together that we might see recurring in numerous different proteins. So globular proteins will often have multiple different motifs that might be combined together in unique ways in order to create our specific protein. So here are some examples of what those repeated motifs might look like. So of course, for beta sheets, there's actually many different ways that you can put your beta sheets together. So you have a variety of different kinds of beta sheet motifs that you might see. So here's one typical combination up here in the top left. This is the typical way of how you might connect your beta strands together as you're making a beta sheet in a globular protein. We don't commonly see this kind of crossover over here on the right, where you have different strands crossing over each other. And instead, they kind of try to make this more spread out structure. You're kind of alternating which strand is coming in at different times. In general, with our beta strands, right, we're trying to maximize the number of strands that are sitting next to each other. Now, if we're just sitting our beta strands flat right next to each other, right, this is going to lead to a very long structure, right? Again, like we saw in the previous figure, if you're just lining beta sheets up to each other, you can end up with this thing that's thousands of angstroms long, or much longer than your overall protein structure. So we're often going to end up twisting our beta sheets so that we can fit a bigger beta sheet structure within a more compact space. So we can end up with these sort of twisted beta sheets where as we're kind of packing our beta sheets next to each other, they're also turning at the same time to form this twisted structure. And if we twist it far enough, we can actually form this beta barrel where we twist our beta sheet all the way around until it forms a complete ring. So here's a picture on the right of what a beta barrel might look like. So notice we have all of these strands, but they're twisted around to create this overall 3D structure. So you end up with this barrel-like structure, right, with our strong beta sheet extending around the outside. You would have a hollow space down here in the middle, and then, of course, you'd have a space on the outside as well. Beta barrels are actually often used as pores within membranes. So you can stick this beta barrel and use that opening then to allow things to move through a membrane. So you often see beta barrels in membrane proteins, proteins that are within the membrane and then helping to transport things. So the beta barrel is an example of a motif that forms with just our beta strand secondary structure. A number of our motifs will involve both beta sheets and alpha helices. So our beta alpha beta loop is an example of a motif that combines our beta strand together with an alpha helix into this characteristic loop structure. So here we have a parallel beta sheet. But of course, when you have a parallel beta sheet, we need our amino acid strand to get around back to the far end of our other beta strand to then form that parallel connection here. So as our loop goes out, we then form an alpha helix before coming back around to form the next strand of our beta sheet here. So we can then use these beta alpha beta loops in a variety of different formations. We can even combine multiple beta alpha beta loops together, and we can make this alpha beta barrel here, right? So notice how we have now an inner layer of beta sheets, and here it's essentially a twisted beta sheet, almost like our beta barrel. And we have our alpha helices here on the outside, kind of surrounding it in this barrel-like structure. This sort of structure can also be used, right, to help shield those hydrophobic amino acids, right? So you might have more hydrophobic amino acids on the inside here on those beta sheets. And then you might have more of your hydrophilic or polar amino acids on the outside on these alpha helices as you're trying to, again, hide those hydrophobic amino acids from our aqueous solvent. So it does often require multiple layers here in order to really hide those internal amino acids from our solvent there, right? Because the solvent can potentially penetrate through if you don't have a too much structure there to kind of hold that water out. While motifs are one useful way of thinking about our globular protein structure, another useful way of thinking about it is through protein domains. So domains look at structure from more of a functional perspective than just a perspective of looking at what structural elements are there. So a domain is a unit, an individual subunit of a protein that has a particular function, right? So in this case, we have a protein here, and this tan-colored region is a domain that specifically binds to calcium, right? So it's a specific structure that might be conserved, right? Similar different proteins will use similar structures for a similar function, right? So we can see this domain kind of repeated within different proteins that have a similar need, for example, in this case, to bind calcium, right? So you might see a calcium binding domain that's then used within several different proteins. So again, it will have a characteristic shape to it, but it's more defined based on its function and less so its shape. Now, for some of our globular proteins, we might not have a defined protein structure for our entire protein. So these are referred to as intrinsically disordered proteins. So these are proteins that are either lack a definable structure in general or might have certain regions that lack a definable structure. And so what I mean by that is that they don't adopt just one specific shape, but they might be more flexible in changing shape. They would have more of a random coil structure, we would say, where it can kind of rearrange itself randomly. Now, when we talked about secondary structure, right, we talked about some of the amino acids that make it harder to form certain secondary structures. So our intrinsically disordered proteins will tend to have higher concentrations of those sorts of amino acids, right? So things like our helix breakers, our proline and glycine, but then also things like high concentrations of charged amino acids, our lysine, our arginine, and our glutamate. So all these make it harder to stay in a tighter structure, and they end up giving that more extended and flexible structure. Intrinsically disordered proteins can be very useful because of that flexibility. So here's an example. This is the P53 protein. And this protein has multiple different protein targets that it needs to bind to. So this figure down here gives us the Ponder score. So this is a score that just indicates the overall disorder of different regions within our protein. So a high Ponder score indicates a region that tends to be more disordered, and a low Ponder score indicates a region that is more ordered. So notice we have this region at the front. This is a region near our N-terminus that is more disordered and would adopt a more random configuration. And we have this region near our C-terminus that also is more disordered as well. So notice in our picture here, we have a defined structure that we were able to measure for one portion of the protein, but our N-terminus and our C-terminus don't have that defined structure. Right? It's actually not even able to be measured because it would continue moving as you're trying to do your measurement. Now, in this case, our C-terminus actually uses this disorder to interact with multiple different proteins. So when we add different protein targets, the structure of the C-terminus will actually change in order to help it to bind to that particular target, right? So notice it adopts one shape when it binds to this cyclin A protein. It adopts a slightly different shape as it binds to this sirtuin protein, and so on and so forth, right? It's actually able to change its shape as it needs to bond to different proteins, rather than just having one specific shape that can only bond to one specific protein. So there are some advantages to this intrinsically disordered nature, but of course, it's not something that we want for every protein. So as we see here, our protein tertiary structure can be highly variable and highly complex depending on what our amino acid side chains look like. Again, if you have hundreds of different amino acids, you have hundreds of different possibilities for what that tertiary structure could look like. That protein tertiary structure is going to be directly related to the actual function of our protein, right? It might fold into that longer, more extended structure for our structural proteins, and we stay in a more compact shape for our globular proteins. As we see here, we also want to have a shape that is dependent on what binding partners we have, right? You might have one constant shape if you have one key binding partner, or you might be able to change between different shapes if you have multiple binding partners. But in every case, it's the structure that's determining the ultimate function of that protein.